evolutionary change is not always incremental. Presumably, during the microevolution of each species, selection can act on the ecological phenotype in a way that is reminiscent of classical Darwinian selection. It can generate incremental morphological changes from generation to generation. But that does not mean that the generative phenotype is directly comparable between related species that have been separated for a significant number of generations. The gene pools of those separate species, the collective set of genetic determinants that are unique to that species, will have changed in a variety of ways. This results in divergent generative networks of genes that interact in a variety of ways to produce a variety of ecological phenotypes. When layers of complexity are added to previous ones, we can say that it's the creative force of the environment that's responsible. Certainly, the environment plays an essential role, but without the molecular, cellular, and developmental capabilities of cells, none of this could happen. Not only that, and here's an important point for Evo-Devo, without development of multicellular organisms, evolution would be limited by significant constraints. It would not be possible to build new complexity on top of the old, or to vary it to any considerable degree, because that complexity depends on the interactive and specialized capabilities of cells. Incremental changes in ecological phenotypes do not map directly or in a simple way to incremental changes in the generative phenotypic toolkit. Toolkit elements are used and reused, and the ways that they are used are varied. Some of this variation results from regulation of gene expression. Changes in morphology are aspects of the ecological phenotype, but the cellular and molecular wherewithal to create that morphological diversity and complexity we have defined as the generative phenotype. Evo-Devo is a logical outcome of the new insights that are obtained when developmental biology, now greatly improved by empirical advances in molecular and cellular techniques and discoveries, is brought together with evolutionary science. When sophisticated comparisons of genomes obtained via state-of-the-art high-throughput sequencing and comparisons are brought together with visualization of morphogens and deep understanding of the complexities of interacting transcription factors and the like. Broadly speaking, the genotypic gestalt creates the toolkit of the generative phenotype, and the ways that toolkit elements are deployed creates the ecological phenotype. Chapter 14. Contrasting the 19th century concept of incremental change with 21st century evolutionary developmental biology. The terms macroevolution and microevolution were not coined until 1927 by Yuri Filipchenko. Nevertheless, we can safely assume that for Charles Darwin, what was later referred to as microevolution referred to the mechanism of natural selection as incremental changes brought about by repeated cycles of variation and selection in successive generations. Macroevolution would be seen as the long-term result of those incremental changes. From a 21st century point of view, however, we need a conceptual framework that describes the relationship between natural selection and the generation of complex organization from a molecular, cellular, and developmental point of view. To say that incremental change is responsible for the generation of complex organization is not enough. This is an incomplete description. Today, we know far more about the inner workings of cells and the ways that cells interact during the process of development than Darwin could possibly know. Therefore, we are in a much better position to describe the evolution of complex shape and form in a detailed and meaningful way in terms of concrete biological mechanisms. In other words, 21st century evolutionary theory provides a more satisfying explanation than classical Darwinian theory or the modern synthesis. However, to this day, non-scientists are more familiar with classical Darwinian theory and the modern synthesis than with the fascinating principles of molecular, cellular, and developmental biology.